Dr. Hansen here. Today I'll be talking about glycolysis, glucose breakdown. We're talking about oxidizing glucose in order to get energy to live. And of course, half the energy which you take in your diet is in the form of glucose, in the form of starches. Before listening to this talk, I really hope you understood the pre-matriculation videos on biochemical nomenclature and on coenzymes, because if you understood the biochemical nomenclature talk, then every enzyme I talk about today, you will be able to interpret it rather than to just memorize it through brute force as gibberish, which means the entire video today will be a complete waste of your time. I also hope that you've worked through the coenzymes video because most glycolytic enzymes have placed coenzymes at the active site, and it is those coenzymes that are involved in the catalysis. Furthermore, this talk builds upon the carbohydrate talk, where I talked about the structure of glucose and of fructose, and upon the why we breathe talk, where I talked about oxidation reduction reactions. So again, if you've worked through all this and kind of understand it, I think you're in great shape for today's talk. And for today, I want you to be able to look at the glycolytic pathway, interpret each of the enzymes involved, and tell me whether or not each of the substrates and products are high or low in energy. To decide whether you're ready to go through this glycolysis video, or whether you really want to go back and review some of the pre-matriculation material that you didn't understand so well, I would like you to take a look at this list of enzymes. I think you should be able to interpret each chunk of each of these enzymes to write down what the function is overall. Please take a stab at this list of enzymes and at this list of enzymes, which will underlie the next several metabolism talks. I honestly think that this should be relatively straightforward, and you really want to pull this out of your mind before you go forward, since it's going to be a lot easier to interpret enzyme names than it will be to memorize them through brute force. But if you're simply looking at the name and trying to memorize the corresponding definition, you're just wasting your time. The overall pathway of glycolysis is described very nicely in the online textbook, and their description is from a very different perspective than mine, and having both perspectives could be quite helpful in understanding this process that is so central to life. But first, let's very quickly review the why do we breathe talk. What fuel is being burned here? What does the word burn mean? Please answer. Okay, but I'm going to move on. Burning is oxidation. That fire was burning primarily glucose in the form of cellulose, such that the glucose would combine with oxygen so that every carbon and hydrogen atom would be completely bonded to oxygen. And as the electrons fall down in energy from the carbon to carbon bonds and carbon to hydrogen bonds to form oxygen to carbon bonds and hydrogen to carbon bonds, about 686 kilocalories of energy was released per mole. And of course, in glycolysis, as we're talking about oxidizing glucose in the body, we essentially do the exact same reaction, except now the amount of heat released is 326 kilocalories. When you burn the sugar that's in your diet, about half of that energy is released in the form of heat. It can keep your body warm. What happens to the other half of that energy? Please pause and answer. It's very important. The rest of that energy is captured in a step-by-step process, and it's harvested in order to make ATP out of inorganic phosphate and ADP. And we're going to talk about how glucose can be used to do that. We'll also talk about how lipids can be used to make ATP. Whenever I say you're making energy, I mean you're phosphorylating ADP to make ATP. And if we look at the overall process by which glucose gets oxidized down to carbon dioxide and to water, it will begin with glycolysis, the oxidation of glucose to make pyruvate. In turn, the pyruvate will be oxidized to produce acetyl coenzyme A and carbon dioxide, and then acetyl coenzyme A can enter the citric acid cycle, which will further burn these carbons and release them as carbon dioxide, while making high energy electrons to dump into oxidative phosphorylation. Those electrons will fall all the way down onto molecular oxygen to make water. The oxidation of glucose to pyruvate makes 2 ATP, and that is a learning objective. It's a simple enough number, and it's kind of important to realize that you get 2 ATP from that. You should also be aware that from the rest of the process, going from pyruvate down to carbon dioxide and water, you get about 30 ATP. That first part, glucose to pyruvate, which is called glycolysis, can be done anaerobically. In turn, pyruvate can be oxidized to enter the citric acid cycle and oxidative phosphorylation to complete the oxidation of glucose all the way down to CO2 and water, in which case it is called aerobic because you need air. We will be looking at anaerobic and aerobic metabolism in the human body, both in neuromuscular, with respect to how different muscle fibers use anaerobic and aerobic metabolism. We'll also be looking at that in more detail during hematology because red blood cells are exclusively anaerobic. For your information, anaerobic metabolism in red blood cells and in muscle can only burn glucose, no other fuel. Today, of course, I am focused on the fuel glucose and its high energy electrons, which can be dumped into oxygen in order to make ATP. But I want to point out that other fuels can also be oxidized. Lipids and amino acids both have high energy electrons in carbon to carbon and carbon to hydrogen bonds, which can fall toward molecular oxygen in order to be burned. In particular, triacylglycerols can be oxidized and enter the citric acid cycle through acetyl coenzyme A. Amino acids can be deaminated to make keto acids, and again, enter the citric acid cycle as acetyl coenzyme A, or directly enter the citric acid cycle through keto acid intermediates. In fact, if you worked through my pre-matriculation video on amino acids, this conversion of an amino acid to a keto acid should seem very trivial to you, and it should seem easy how to get from, say, alanine to pyruvate. Of course, I can also frame glycolysis, the oxidation of glucose to pyruvate, in the broader context of the central metabolic pathway figure from the Lippincott textbook, which is available online, and I've described this some in the coenzyme pre-matriculation video. And again, I want to remind you that you should not know every reaction in this pathway or every arrow, but as we talk about each of the pathway individually over the next year, I'm going to frame them in the same context. And so we can begin by looking at the sugars, and we can even zoom in specifically on glycolysis. And as I mentioned, glucose lysis, glucose is the substrate of glycolysis, and the end product of glycolysis is pyruvate, and it gets oxidized. But will you pause the video and confirm that in fact this was an oxidation, that glucose went down in energy to pyruvate, and then I will describe how that energy is used to make ATP. Please pause. This is fairly simple. All you have to do is look at the number of carbon atoms. We know glucose has 6, 1, 2, 3, 4, 5, 6, and pyruvate has 3, 1, 2, 3. The hexose is cleaved into 2 trioses, but you have the same number of carbon in all. Then we look at the number of carbon to oxygen bonds. 1, 2, 3, 4, 5, 6, 7 carbon to oxygen bonds. And we look in pyruvate. 1, 2, 3, 4, 5, 10. So we went from 7 bonds to oxygen to 10 bonds to oxygen. So glucose absolutely was oxidized in order to become 2 pyruvate molecules. Before I zoom in and look at each of the reactions which occurs in the oxidation of glucose to pyruvate, I want to further set the context, since this is half of all the energy which drives your life. How is it that glucose gets into a cell to begin with so that it can be used as a fuel? Please answer the question in general and then give me as detailed of an answer as you can to describe the mechanism by which glucose enters cells. But I need to move on. Well, of course, glucose is way too hydrophilic to diffuse through a membrane. Therefore, it needs a channel. In particular, there are several types of channels, but I want to focus on the glucose transporters, GLUTs. Absolutely a learning objective. You need to know about these. GLUTs do not require any energy. Rather, what drives the transport of glucose through a GLUT is going to be a concentration gradient. Generally, you eat a meal and you have a high blood sugar level. The glucose then will bind to one side of the transporter and it will interconvert to a different conformation to be released on the inside of the cell. If there were a high concentration of glucose on the inside, it would likewise be driven back out by a concentration gradient. Of course, the question then is, can you tell me how glucose gets trapped inside of a cell? If you know the answer, try to answer it in detail. If you don't know the answer, at least take a stab at it. Well, glucose gets modified. In particular, the critical enzyme, which is a learning objective, is hexokinase. Please interpret the name to tell me what it does, what its substrate is, and what its product is. A kinase is one of the most important learning objectives from the pre-matriculation video on biochemical nomenclature. And you're going to run into a dozen different types of kinases, many of which can have loss of function mutations and lead to genetic diseases. Hexokinase, shortened from hexose kinase, is an enzyme that adds a phosphate group to a hexose, a 6-carbon atom sugar. Hexokinase is considered the first step in glycolysis because glucose 6-phosphate can go through glycolysis. However, hexokinase is not the committed step in glycolysis because glucose 6-phosphate can also end up going through the pentose phosphate pathway or through glycogen synthesis, as we'll describe in detail shortly. Glucose enters a cell and then it gets phosphorylated by hexokinase. Wait a minute. Where does hexokinase get a phosphate group to add to the glucose? Please pause and answer that question. Again, that was clearly described in the pre-matriculation talk on coenzymes. Of course, the primary source of high-energy phosphates in the cell is going to be ATP. So hexokinase grabs glucose, it grabs the coenzyme ATP, and it positions the glucose to nucleophilically attack ATP to pick up a phosphate group. Can you verify that that phosphate is attached to carbon 6? Also, will you go ahead and draw the arrows to show how that nucleophilic attack occurs? And for that matter, will you label each of the substrates and products as to whether they are high energy or low energy? Please pause and do so. I'm hoping that these questions do seem simple to you because we're going to do them over and over and over all year long, the exact same reactions, just with different names of enzymes, substrates, and products. In fact, if you find these 3 questions to be strenuous and you feel you have to think a lot to do them, please give me a phone call. I think that a very short explanation from me would be an extremely efficient use of your time, because if you understand the principle, you will then very easily be able to apply it over and over and over instead of each time you're running into it, depending upon just having memorized something that's equivalent to gibberish. And so the numbering from the carbohydrate presentation, anomeric carbon, 1, 2, 3, 4, 5, 6. And so this is glucose 6-phosphate. In terms of the nucleophilic attack, can you see where to draw the arrow showing the pair of electrons attacking the electrophile in the double bond, and how to have the double bond resonate up to form an oxyanion? A lone pair of electrons on O6 of glucose nucleophilically attacks the phosphorus atom of the gamma phosphate, pushing a pair of electrons in the double bond up onto the oxygen to make an oxyanion. Then in this intermediate, the electrons resonate back down from the oxyanion to reform the double bond, kicking off the bond between the phosphorus atom and the linking oxygen in order to push off ADP, leaving the gamma phosphate now linked to glucose to make glucose 6-phosphate. And I'm hoping that's simple. In terms of energy, in going from ATP to ADP, there was this huge compressed negative charged region. When you kick off the phosphate, it allows those negative charges to separate, so ATP goes way down in energy when it loses a phosphate. You could call this hydrolysis, but it wasn't water that attacked it. It was O6 of glucose. And the glucose went up in energy because having that charged phosphate group linked to it decreases entropy and makes a more complicated molecule. Were you to nucleophilically attack this phosphate group now and release inorganic phosphate, there would be more free resonance around the linking oxygen, leading to lower energy. So we have glucose 6-phosphate. Why can't that escape the cell just as easily as glucose can? Well, the obvious difference is it has this large, negatively charged, bulky group. That phosphate must interfere with the transporter. Whereas glucose will go from high to low concentration, so if it's high outside of the cell, it'll go in. If it's high inside of the cell, it will go out. However, when you add a phosphate group to glucose, it no longer can cause the transporter to flip from the open inside to open outside. Therefore, glucose 6-phosphate just is too bulky to get through the hole. At the same time, there are cells which need to export glucose. Do you know what type of cell that is? Please see if you can think for a minute what type of cell might want to export glucose. And of course, if most of glucose in the cell is in the glucose 6-phosphate form, how do you export it? Please pause and try to answer that. Liver cells specifically need to export glucose. Liver cells store glucose in the form of glycogen during times of plenty, and then when blood glucose is low, the liver cells tear the glycogen apart and release the glucose to replenish the blood glucose supply. Also, kidney cells need to export glucose, and that's because they recover glucose that might be lost through filtration. Of course, I just got through saying that most of the glucose in the cell is in the form of glucose 6-phosphate, and you can't export that. So how do you release it? Well, you need to break the phosphate back off to regenerate free glucose, just glucose. And the enzyme that you need to know that does that is glucose 6-phosphatase. In fact, this is a very important phosphatase that you do need to be familiar with, because in the future, we will learn how a loss-of-function mutation of glucose 6-phosphatase can lead to a severe metabolic disease, glycogen storage disease type 1. Does a cell have to spend energy in order to break that phosphate off of glucose 6-phosphate to regenerate glucose and then it can be released from the cell? Please pause and answer. Pretty much by definition, hydrolysis reactions are going to go down in energy because they're oxidations. The oxygen atom in the water ultimately ends up as part of the phosphate. In addition, in this case, however, also you've gone from one complicated molecule to two simpler molecules, so entropy has increased. Furthermore, the electrons in inorganic phosphate can resonate more freely among all four oxygens than they can when one of the oxygen is in a linking bond to a compound such as glucose. So absolutely this goes down in energy. Hydrolysis of phosphates are always going to be spontaneous. Can you draw the nucleophilic attack step? Please draw a couple arrows. Again, look for the double bond. Lone pair of electrons on the oxygen nucleophilically attack the phosphorus, resonates up onto the oxygen, resonates back down, and kicks off the glucose. I now want to look at the second step in glycolysis, where glucose 6-phosphate is not only trapped inside of a cell, but now it can freely interconvert between a glucose 1-phosphate form in a fructose 6-phosphate form. The enzymes which do this are called phosphoglucomutase and phosphohexosisomerase. As we described in the pre-matriculation videos, it is a learning objective to know that an isomerase simply changes the isomer of a compound. That is, you have the same number of bonds, the same types of bonds, but they're organized differently. For example, here the phosphate has been moved from carbon 6 of glucose to carbon 1 of glucose. It is also a learning objective to know that a mutase is simply a type of isomerase. It's also a learning objective to know that since the substrates and products of isomerase reactions all have the same number of bonds and the same types of bonds, the relative energy between these substrates and products are about the same. Based on that knowledge, what do you predict will be the relative concentrations among the various phosphohexoses shown here within each of your cells? Please pause and answer. Since each of these 3 hexose phosphates has similar energy, and since you have enzymes which lower the energy barrier between them, they will freely interconvert, flow back and forth, such that in most of your cells there will be an approximately equal concentration of all 3 of them. Please do not put a lot of intellectual effort into memorizing whether phosphoglucomutase interconverts glucose 1-phosphate to glucose 6-phosphate, or whether it's phosphohexosisomerase that does that, or which interconverts between glucose 6-phosphate and fructose 6-phosphate. I would just look at them as essentially equivalent because you have more important things to memorize. They're both isomerases. They're both about equal in energy. Nonetheless, just for your information, phosphoglucomutase interconverts between 2 forms of glucose, gluco, whereas phosphohexosisomerase refers to 2 different types of hexoses, glucose and fructose, in case you're curious. Let's put this in the context of the central metabolic pathways figure. So we've shown that glucose can enter the cell, get phosphorylated to glucose 6-phosphate, and then it will freely flow back and forth between glucose 6-phosphate, glucose 1-phosphate, and fructose 6-phosphate. They will then be freely available to enter several different metabolic pathways. Glucose 1-phosphate can enter glycogen synthesis. Glucose 6-phosphate can enter the pentose phosphate pathway, and fructose 6-phosphate can enter glycolysis. How does a cell regulate whether glucose is going to be burned through glycolysis in order to produce energy, make ATP and NADH, or whether glucose will be assembled into a polymer by glycogen synthase for fuel storage, or whether it will be used by the pentose phosphate pathway in biosynthesis in order to make ribose and in order to produce the biosynthetic coenzyme NADPH. Regulatory steps require a drop in energy, and I already mentioned that isomerases do not lead to changes in energy. To more graphically see how each of these pathways can be regulated by enzymes at crucial energetic metabolic steps, let's take a look at glycolysis with an energy landscape figure. This energy landscape figure is drawn to parallel a hill with a flat top. On the flat top of the hill, we have glucose 1-phosphate, glucose 6-phosphate, and fructose 6-phosphate because these 3 phosphohexoses are all pretty equal in energy, and it's easy to push them back and forth between themselves. They can flow freely. Here, phosphofructokinase 1 grabs the substrate fructose 6-phosphate and adds a phosphate to it to form fructose 1,6-bisphosphate. So the sugar has gone up in energy. Now, why is it a regulated step? Because the net energy change is negative. That's because the phosphate that is added to fructose 6-phosphate comes from an even higher energy source, ATP. The phosphofructokinase binds fructose 6-phosphate and ATP, positions them for a nucleophilic attack, so the phosphate gets transferred from the ATP to the fructose to make fructose 1,6-bisphosphate a high-energy sugar. In the meantime, ATP has plummeted in energy to ADP so that this step is irreversible. And that's analogous to taking a rock on the top of the hill, which can be pushed back and forth, but if you push it over the edge, it plummets, it's going to stay at the bottom, and that is why phosphofructokinase 1 is the key regulator of glycolysis, as explained here. And in fact, this same idea of the net energy change making these steps irreversible applies to the pentose phosphate pathway and to glycogen synthesis. A learning objective is to identify the key regulator for glycogen synthesis, pentose phosphate pathway, and glycolysis. Please go ahead and do so, but I'm going to move on. Well, of course, glycogen synthesis is regulated by glycogen synthase, and the reaction has a net negative energy. Pentose phosphate pathway is regulated by glucose 6-phosphate dehydrogenase, which again has a net negative energy. And, as we mentioned already, glycolysis is regulated by phosphofructokinase 1, which again plummets in energy, making it irreversible. Looking again at glycolysis, I want to clarify the difference between regulation and a key committed step in regulation. Of course, phosphohexoses are formed by hexokinase and isomerases. Accordingly, hexokinase does regulate glycolysis because if hexokinase increases the amount of the substrate fructose 6-phosphate, that'll increase the rate of glycolysis, and if hexokinase produces less of the substrate fructose 6-phosphate, that will decrease the rate of glycolysis. However, hexokinase is not the committed step in glycolysis because the substrate fructose 6-phosphate can ultimately enter the pentose phosphate pathway or glycogen synthesis. The committed step, the key regulated step of glycolysis, is phosphofructokinase 1 because that catalyzes the formation of fructose 1,6-bisphosphate, which then is committed to glycolysis. In other words, just as you could say that hexokinase regulates glycolysis, it also regulates the pentose phosphate pathway and glycogen synthesis, but again, it's not the committed step for any of these pathways. It can upregulate or downregulate all of them to some extent. There are much more powerful and much more important regulators of these pathways. To better understand this type of regulation in the case of glycolysis, let's consider when either insulin or glucagon affects the activity of phosphofructokinase 1. For example, let's say that we inhibit phosphofructokinase 1. How will that downregulation affect glycolysis, the pentose phosphate pathway, and glycogen synthesis? Think about the dam model and explain it. When phosphofructokinase 1 is inhibited, much like water going over a dam, the upstream substrates, fructose 6-phosphate, will build up, whereas the downstream products, fructose 1,6-bisphosphate, will be decreased because you're not making them as much. Now, how will that affect the concentration of glucose 1-phosphate and glucose 6-phosphate? Please answer. Of course, if fructose 6-phosphate has built up, Very quickly, it's going to reach equilibrium with glucose 6-phosphate and glucose 1-phosphate so that all are about equal concentration. Accordingly, inhibition of phosphofructokinase 1 will lead to an increase of all three of the phosphohexoses. How then will that affect the rate of the pentose phosphate pathway in glycogen synthesis? Well, since you have more substrates, there will be a slight increase in these other pathways. Now, instead of inhibiting phosphofructokinase 1, what if you stimulate it? How will that affect the pentose phosphate in glycogen synthesis? Please pause and answer. As you speed up glycolysis, the amount of the substrate fructose 6-phosphate will now be depleted, and it's in equilibrium with glucose 1 and glucose 6-phosphate, so that all these substrates will equally be decreased, and thereby the glycogen synthesis and pentose phosphate pathways will also be decreased. I'm not going to describe how inhibiting or stimulating glycogen synthase will affect all these other pathways, but I think it's in your interest to kind of go through that quickly to practice the DAM model algorithm and to be sure you understand how to think about these enzymes and pathways. And likewise, I suggest you do the same thing for inhibiting or stimulating glucose 6-phosphate dehydrogenase. Of course, as you look at these pathways on your own, don't hesitate to contact me if you want to confirm any of your understandings or if you find any part confusing. But now I want to show these three pathways in the context of the central metabolic pathway figure that's in the Lippincott textbook. Don't be afraid to read this part of the Lippincott textbook also. But again, we've been talking about glycolysis, glycogen synthesis, and the pentose phosphate pathway in looking at the context. You'll notice that all the carbohydrates in this chart are in blue, the lipids are in brown, and the amino acids are in green. But we're focused today on the carbohydrates and specifically on glycolysis. So now let's zoom into that. Of course, we've already looked at the first few steps, hexokinase, phosphohexosisomerase, and phosphofructokinase. These first three steps, which involve hexoses, are considered the preparatory steps, and the very next step, the hexoses, are cleaved into trioses. So the rest is considered a triose part, and in particular, this is where the energy is harvested. Now my plan is to look in detail at every one of the glycolytic enzymes. But before I do that, I want to point out that most of the glycolytic enzymes also play a role in gluconeogenesis. So here on the left side of the figure, glucose gets oxidized to pyruvate. On the right side of the figure, we can see that most of the same enzymes are involved in regenerating glucose from pyruvate. So in part 2, which is about gluconeogenesis, I will begin with glycolysis because so many of the enzymes are the same. Before going on to gluconeogenesis, if you have any questions about this, please don't hesitate to contact me. Focus on first step, final step, and regulation in all of these pathways as we look at central metabolism. Thank you.